This recorded session is dedicated to Module 5. Our topic is lymphocytes. So we're now going to talk about B cells and T cells in detail. So at the end of this module, you are expected to be able to identify the different steps in the development of B cells and T cells. And at the same time, also, you must understand the differences on the development, action, and responses of both types of lymphocytes. So, as a quick review, we are reminded of the significance of lymphocytes, both T and B, on the process of specific immune recognition. Actually, not only on the recognition part, no, but also with the mounting of the immune response. We know that adaptive immune systems or the adaptive immune system, yung section natin where memory recognition are vital components. No? Um, the lymphocytes, both and B and T, are very much involved. In terms of origin of these two lymphocytes, all lymphocytes are derived from the bone marrow. You know? Though their maturation sites are of varying nature. So for T lymphocytes, the maturation part or the organ where we expect them to mature is the thymus, while for the B cells naman, for the B lymphocytes, it's still in the bone marrow. Now, both B and T lymphocytes, they are derived from the small lymphocyte progenitor, which is derived from the common lymphoid progenitor of the multipotential hematopoietic stem cell or the HSC. No? Mature forms of T lymphocytes and B lymphocytes, especially those that are ready to develop antigen specificity, they're found mostly in secondary lymphoid tissues and organs. So in our lecture, we're going to actually work on or discuss more on the biology of both B cells and T cells. Kasi these or introductory topics like where they are found, where do they mature, diba? Napag-usapan naman natin yan during the previous modules. But I think no, we just have to reiterate again. Okay, so now let's begin our discussion with B lymphocytes. So B lymphocytes, they primarily interact with foreign antigens in order to do several things. No? So their interaction with foreign antigens either is important for immunosurveillance. No? After your antigens interact with your B lymphocytes, they may differentiate as memory cells. So important for immunosurveillance, and also they may differentiate into plasma cells, also for immune for what they call this, for antibody secretion, for antibody production. Diba? So those are the two fates of naive B lymphocytes after they interact with a specific antigen, after they interact with a specific foreign pathogen. Immunosurveillance, as a review, it's an all-encompassing term. Diba? So we define immunosurveillance as a process by which immunologic cells look for and recognize. Okay, so there's active searching and there's specific recognition of foreign pathogens such as um, bacteria, parasites, viruses, and within us, mga ano natin, no, those cells that are precancerous or are already cancerous. On the other hand, antibody production is an activity that's mostly carried out by the B cells that differentiated into plasma cells. We remember from module number 4, that antibodies are protective proteins that are produced by our body in response to the presence of a foreign substance or a foreign cell. Okay, so the mechanistic association between antibodies and antigens, we are able to we're able to appreciate that, no, as as a phenomena that's very specific, or a phenomenon that's specific to the molecule level. So we expect yung ano nga, 'di ba, yung complementarity, the concept of epitope paratope complementarity. Okay? So, um, um, when we talk about, or when we refer to the the specific interaction of antigens and antibodies, diba? We are reminded of their physical association. So antibodies, they recognize antigens, they latch onto antigens in order to remove them from the body, like kung circulating yung antibodies, no, so that the antigens are deactivated, or could be that antibodies cover or latch onto the antigens that are presented on the surface of a pathogen, no, so that the pathogen will be flagged or will be marked. No? A wide range of substances are regarded as antigens. As a review, it's not only proteins that are secreted or proteins that are present on a pathogen's cell surface that can be considered as antigens that antibodies recognize and bind into. No? There are also other antigens such as toxic materials, mga toxins such as insect venom. So we discussed antibodies that aside from the yun nga, no, circulating ones na we expect to really bind to antigens actively. We also discuss antibodies that are also membrane bound and do not really contribute to the titer, to the circulating titer, the circulating concentration of antibodies in our blood. So these membrane-bound antibodies, they are dedicated for recognition and interaction with antigen plus the cell where the antibody or the B cell receptor is bound into. Also in B cells, diba? When as they mature, or in the context of a group of B cells that are maturing as one clonal population, you expect them to have the same membrane-bound antibody, the same B cell receptor in terms of nature and behavior, diba? So we have to remember that na during clonal expansion, a group of B cells that are activated by the same antigen, activated by the same foreign material, they will undergo the same clonal expansion processes. Same clone, they will have the same signaling. They will have the same set of signals. Therefore, yung B cell receptor, yung membrane-bound antibody that they will develop, okay, is expected to be of the same nature. Now, let's have a quick review of the anatomy of immunoglobulins. Diba? So you would remember that there are two major regions of immunoglobulins that are attached onto a B cell. So yung B cell receptor natin is divided into two regions. We have the fragment antibody binding region, which is where the antigen recognition site is found, and then we have the fragment crystallizable region, for where we expect it will have a different I don't know it will have different components based on the antibody type, and um, it will have a variety of effector functions, effector mechanisms na maaring ma-activate. No? So for membrane bound, immunoglobulin, a BCR, no, we already discussed about associated proteins, yung alpha and beta proteins natin that actually that again as a review, no, facilitate the conduct of signal from an antigen-bound B cell receptor. So once that an antigen binds to the antigen-specific recognition site of our antibody, then signal will travel throughout the B cell receptor that we expect, no, immunoglobulin beta alpha proteins associated with the B cell receptor to ensure the downstream flow of signal. No? Now, since we've mentioned about antigen exposure or antigen binding to our B cell receptor, it's important that we also uh, visualize, how uh, antigen binding, antigen exposure really really is involved in B cell biogenesis, and also the how antibodies are formed for the case of plasma cells. So here, so antigen exposure is really a pivotal or a key event in the lifetime of these cells because it triggers differentiation to either the plasma group or the memory group. No? So if you're going to trace yung history, yung maturation history, so from the, 'di ba, from the stem cell, you really expect dedicated pool of cells to differentiate into pre B cells. Then you will have your antigen specific or your B cells that have matured, considered still naive, no, but still wala pang activation because antigens are not yet present. So once that these naive mature B cells having their receptors, no, are already able to what do you call this already able to what do you call this already able to uh, expose themselves or bound themselves into antigens, no, that are from the external environment, and the maturation, the fates of your B cell will now be indicated. Either they will be antibody secreting plasma, or they will be memory cells. Okay? And then, aside from B cell receptors pala, no, there are other cell surface receptors that you expect to find in a B cell, ano, in a uh, 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 differentiating B cell, to be specific. So these receptors, no, they usually appear from ground up. Ibig sabihin, there's no need for them to really be synthesized from an original scaffold. So they appear de novo. So they appear na parang once that your B cell differentiates, as it differentiates, it will just acquire these cell surface proteins because they're specific for, or not specific, but they are involved in how the B cell will mature, how will it behave. No? So one surface protein of B cells, aside from the B cell receptor, is the MHC class 2 receptor. So MHC class 2, they are expressed only in aside from B cells, and they are expressed in dendritic cells, in macrophages, and other immune cells. No? So MHC class 2 molecules, they display peptides to the CD4 plus lymphocytes in order to activate the humoral mediated immune response. Okay? And we're going to have a dedicated discussion about about MHC in the latter modules. But the thing is, it functions for antigen presentation. Aside from MHC class 2, another cell surface protein is specific, or is that you expect to find in a B cell, is the B7-1 and B7-2. So B7 is actually a type of peripheral membrane protein. So it's a membrane protein we expect to find on activated antigen presenting cells. Okay, so binding of the B7 to of an antigen presenting cell to a T cell will cause inhibition of the activity of activity of some T cells, or also their stimulation. So it serves as a suppressor. Then we also have CD40, which is expressed not only in B cells. No, it's actually it can be found also in dendritic cells, in macrophages, in some instances, vascular cells, or blood vessel cells, endothelial cells and smooth muscle cells, fibroblasts, and epithelial. No? So CD40 stands for cluster of differentiation 40. So it's a co-stimulatory protein that is found on antigen-presenting cells and is also required for further activation. And then we also have CD19, no? Which is what we call the B cell surface antigen. So specific siya talaga for B cells. Ito. B cell um, B cell marker siya, CD19. So also called B lymphocyte surface antigen. Okay? And then mitogen receptors, so receptors that are capable of recognizing mitogens. A mitogen is a compound, a small protein or a peptide that can induce a cell to do cell division. So B cells have mitogen receptors. So they're sensitive, or they are capable of recognizing when the environment is rich with cell dividing or cell division stimulating compounds. Okay? All right. So now, moving forward, let's go back to our discussion of B cell maturation. Say we mentioned a bit about its, membrane biology. Okay. So here, a naive B cell is a mature B cell. However, it has not encountered any antigen yet, and also in terms of the B cell receptor. Okay. So it expresses both IgM and IgD in their as their possible as their BCRs. Primarily the what do you call this, the cycle, or not really cycle, but the alternative between IgM and IgD is because of alternative splicing in the genetic component part, no, prior to that, yung ano pa, yung mismong genes that are coding for the receptors for the IGs, no. So after leaving the bone marrow, so once that they're mature, no, we expect them that some will seed the secondary lymphoid tissues and some will circulate for immunosurveillance. In the sites where they are deposited, these B cells contribute in the immune response that will depend on where the antigen initially entered. So here we can see that the initial site of antigen entry will dictate where the initial site of immune response will be. So if antigen enters through the blood, we expect the lymphocytes to work on it in the spleen. If the antigen is recognized in the skin, then we expect the cutaneous immune system, or the lymph nodes. Once that lymph fluid is drained in the lymph nodes, then in any mucosal surface, the initial site of immune response is always our mucosa-associated lymphoid tissue. Okay. So, now that we have accomplished our review of B cells, pag-usapan natin yung where did they form, bit about their biology, and then yung fates nila, no? We'll also do the same, but now with our T lymphocytes, no? So our T lymphocytes, there are two types that we have mentioned multiple times in the previous modules. So we have our helper cells and our cytotoxic T cells. So as a review, our T lymphocytes, they arise originally from the bone marrow, the common lymphoid progenitor, and they migrate to the thymus in order for them to mature. So during the process, T cells will somatically rearrange their gene segments, or there's genetic rearrangement, eventually leading to the expression of a unique antigen binding molecule, which is the T cell receptor. So, ano to, no? Analogous structure with the B cell receptor. However, with PCR, wala siyang counterpart which is circulating, unlike in B cell. So, you have your circulating Ig. So, for T cell receptor, the biology of the T cell receptor is different from the BCR. So, this receptor, the TCR, allows monitoring of the presence of foreign material, also helps, no, in the destruction of a potentially pathogenic organism that entered the animal system, the human body. Now, let's take a look at helper T cells and cytotoxic T cells further. I think I've also mentioned before, about other types of T cells aside from the cytotoxic and the helper. We'll also include them in our discussion. But let's begin first with our two basic types, no? Our helper T cells and our cytotoxic T cells. So, for helper T cells are one of the most important cells in the adaptive immune system because they are required for almost all types, no, of immune responses that are of adaptive nature. They do not only activate B cells so that B cells secrete antibody or they become, memory cells. They also activate through lymphokines, macrophages in order to destroy ingested microbes for the case of intracellular parasites. They may also activate other types of T cells, mga cytotoxic, in order to kill infected target cells. No? So, the wide or the magnitude of the importance of T cells cannot be or what was essentially better appreciated in cases like AIDS, no, where the lack of T cells really dampened or really degraded yung immunity ng isang individual organism. So for AIDS patient, 'di ba? The reason or the thing is since their T cells, particularly helper T cells, are very low or really the concentration is not if the concentration is not ideal, then the immune response is really very weak, no? So helper T cells themselves can only function when activated to become effector cells. So aside or to now add more details, no? So we have um, effector T cells. Okay, so effector T cells that are of two types. So we're going to discuss them in a little while. Okay, what else? I've mentioned this already. Okay. Mm-hmm. Okay, so now we are now going to proceed with our discussion about effector T cells. So two types of helper T cells. These two types of helper T cells are functionally distinct. Okay, so they're functionally distinct. If the T cell differentiates into a Th1 helper cell, T helper type 1, T cell, what you expect them to do is that they're capable of secreting interferon gamma and tumor necrosis factor alpha. And they can activate macrophages to kill microbes located within phagocytes. Yung intracellular killing, actually Th1 cells facilitate that. So they secrete the important lymphokines, chemokines for that. Then as such, no, they can aside from activating or aside from targeting macrophages, at the same time, Th1 can also what do you call this? Can also activate other cells, especially cytotoxic T cells and B cells. Okay. Now, if the naive T helper cell um, matures into the second type, in Th2. Okay, so Th2, by contrast, it will secrete different kinds or different set of chemokines. So it will secrete interleukins 4, 5, 10, and 13. They are mostly important for extracellular killing, for protection against extracellular pathogens. Also, Th2 type of effector T cell stimulates B cells. No? So, more on the hypersensitivity pala. I think I did not mention this yet. No. So Th2 effector cells are involved in hypersensitivity reaction. They can stimulate B cells to make most classes of aside from stimulating B cells, no, to make antibodies. Though they can also to be specific, they can stimulate IgE. So they can stimulate IgE and some subclasses of IgG that are important. You remember how these two types of immunoglobulins are involved in hypersensitivity, diba? They bind to mast cells, basophils, and eosinophils. So, in cases where the pathogen is really kaya niyang mag-survive on its own without really going inside a macrophage, our Th2 effector helper T cell will be the one responsible. So here, let's take a look. Okay, so the the differentiation of our helper T cells into effector types Th1 or Th2, they will determine the nature of the corresponding adaptive immune responses that these effector cells can carry out, or they can activate. Whether a naive helper T cell becomes a Th1 or a Th2 will depend mainly on the cytokines present when the helper T cell is activated by a mature dendritic cell. So this all happens in a lymphoid organ, a peripheral lymphoid organ. So mature dendritic cells, okay, with their co-stimulatory molecules B7 can activate naive helper T cells either into effector Th1 or effector Th2. And of course, there has to be chemokines present, no? So the difference in the chemokines will actually define whether it will be a naive helper T cell will be an effector Th1 or an effector Th2. Okay? So and then I've mentioned already itong yung subsequent immune responses nila. So remember, the activation or the effector maturation happens in a peripheral lymphoid organ. Okay? So now, let's move on to our discussion about cytotoxic T cells. So cytotoxic T cells, we've discussed, initially introduced them as capable of lysing virally infected cells, and they're also capable of suppressing delayed hypersensitivity. So our cytotoxic T cells, they recognize antigen in the context of MHC class 1. And like our T helper cells, they're fully activated by a set of accessory stimulatory, co-stimulatory molecules or proteins. So ZTLs, they're able to kill target cells directly by activating apoptosis, by inducing apoptosis. And a bit about their biology. So they express the CD8 co-receptor. So our cytotoxic T cell CD8. Okay. Now, so since we've talked about helper T cells and cytotoxic T cells, and I think I've mentioned a lot about cytotoxic T cells, no, in our introductory modules. We're now proceeding with the other functional types or functional groups of T cells. So they include our regulator T cells and our suppressor T cells. Okay, so for our regulator T cells, there are several mechanisms that T regulator cells are capable of carrying out. So as regulatory from the name itself, no, dapat we're already getting what do they do. So aside they mostly work on the activation, deactivation of pathways, unlike our effector helper cells, effector T helper cells, and our cytotoxic T cells that mostly do grunt work, secretion of necessary chemokines and yung direct killing, no, or induction of apoptosis. Our regulator T cells do work on upstream steps, no. So some of the basic mechanisms that T regulator cells use. So we have inhibit or what do they do? So they can inhibit cytokines, or they can inhibit yeah, they can secrete rather inhibitory cytokines so that effector T cells can be deactivated. So if their function is not needed anymore, okay, so inhibitory cytokines are secreted by T regulatory cells, okay, and then also cytolysis, okay, which includes granzymes of different nature, granzyme A or granzyme B. Okay, so they can kill apoptotic effector T cells, mga T cells that are already probably not functioning anymore. So regulator T cells, secrete granzyme in order to induce apoptosis. Then they can also do metabolic disruption, which includes high affinity CD25, okay, CD25 protein. And then normally here we expect uh, uh, several pathways can be activated by the CD25 um, CD25 dependent processes that our T regulator cells work on. And then we also have T regulator cells capable of targeting dendritic cells. So when your T regulator cells active target dendritic cells, no, they can inhibit maturation of effector cells for the T helper group. So you see how more upstream yung function ng regulator T cells. Either they secrete chemokines, they secrete enzymes, or they work on or they have co-stimulatory proteins, or they have receptors responsible for metabolic disruption, and also they can target other activator cells, like for the case of dendritic cells. Okay, so um, in addition to our regulating T cells, we also have our suppressing or T suppressor cells. So a suppressor T cell is a T cell that's capable of inhibiting the activity of B cells and other T cells. So parang similar then with your regulator, but this time it's more of the mechanism, or the expected output is direct inhibition. No, say in regulator there's or the activity is of two-way. Either there's activation or deactivation. No, so uh, suppressor T cells they regulate immune responses, and actually they mediate dominant immunologic tolerance. No? So suppressor T cells also are of MHC class 1 restricted cells. They act on antigen presenting cells by a contact dependent manner, and um, um, they can make helper cells very tolerogenic. No? So T suppressor cells also, in terms of additional description, no, they express the CD8 transmembrane glycoprotein. So CD8 plus sila in terms of recognizing, or in terms of in terms of cell surface protein. So um, when when the invading pathogen is no longer present, so T suppressor cells will now be ready to inhibit all the mounted immune responses. So yun ang ginagawa nila. If the invading pathogen is already destroyed, then they deactivate the immune response. They are the ones responsible of telling the effector T cells to stop the attack, to stop the mounted immune response. Now, in terms of their physiology, they're very sensitive to high concentrations of circulating lymphokine hormones, and they can also release their own lymphokines after the immune response has achieved its goal. So, for sure, or for a fact, suppressor T cell lymphokines are capable of telling T cell effector T cells to stop their immune activity. Now, in terms of memory B cells, even though they're already inhibited by suppressor T cells, some memory B cells will remain after the signal has been after the signal of attack, the signal for immune response to be constitutive. After that signal has been eliminated, some memory B cells will actually remain. So sila na yung remnants of the episode of a particular episode between the effector T cells and the antigen or the foreign pathogen. So they will serve as a memory of how to again carry out yung same response if the pathogen will enter the animal body once again. Okay, so now last two slides no, before I end this first part. So I don't know, I decided to divide this lecture pala into two kasi when I was trying to upload the 2 hour video, almost 2 hour video, it really took more than a day. So I decided to really divide it na lang into two sessions for this discussion content. Because aside from this one, there's also another material, another deck that I will be discussing still about T cells and B cells, but more of magiging review na rin siya since I've mentioned some contents here also. So moving forward. Now, aside from B cells and T cells, diba, you remember from the no, from the um, maturation ng common lymphoid progenitor, we have our what do you call this, our large granular lymphocytes, no? So our large granular lymphocytes, specific example, mga natural killer cells, so they mature alongside with our lymphocytes. So our large granular lymphocytes, no, they're actually mononuclear phagocytes capable of cytotoxic ability, similar with our cytotoxic T cells. They can recognize surface changes in tumor cells and virally infected cells. However, the recognition is not specific, okay, unlike our lymphocytes. So they use non-specific recognition systems. And a good example of this are natural killer cells, which are, along with T cells and B cells, they originate from the common lymphoid progenitor. Now, however, yun nga, they do not use specific recognition systems. And essentially, our natural killer cells, most of the actions of this type of cell has been documented against I don't know, mga tumor cells, no, or cancerous cells in the animal body. Okay, so that this will be the last slide for the first section, and then I will be recording another session. We'll be discussing the lymphocyte activation. Okay.